Hi, everyone. Welcome to our first Fertile Ground Tapas Talk. I am Reynolds. I'm hosting. Today we will have three presentations, not just two, like I said in the newsletter. We'll have three presentations from Isabella and co. I didn't have enough room on the title slide for all your names. Josh and Michelle will be our three presenters. Let's see. So I'm Ryan Knoll, she, her, hers. I'm the curriculum lead for Tiny Earth. Um, Sarah Miller is also here today. Laura Langley might be joining us late. Um, uh, okay, thanks for the heads up in chat. So yeah, we got a lot of great folks here at HQ who help things run. Big shout out as always to M. If anyone's ever used the Discovery database, M's the one who made it. So always like to thank her. Quick announcements from HQ though, before we get started with the presentations. As always, watch the newsletter. All the fun stuff goes there, like this meeting did. Coming up how is it the end of September? Has been the theme of today. Also, what day of the week is it? It's also been the theme of today. October 1st coming up is our mid-semester Discovery Database office hours. We can come chat with me. Em is usually there too, and we can help triage problems or get you set up for success there. And then December 1st is our end of semester one for the folks trying to get isolates into the database before the semester ends or as things are wrapping up. Also, October 1st, watch out for that newsletter. The registration for Tiny Earth and Titletown will open then, which will be hosted in December, on December 10th. That will be in Lambeau Field here in Wisconsin. All this semester, and we plan every semester after that for a while, students are invited to submit their posters or a recording of a talk that they have, or their favorite photo of their favorite isolate, or just a fun thing that grew on a plate that wasn't supposed to. Well, always welcome for students who submit their favorite bit of digital media from their Tiny Earth course to our new Tiny Earth Virtual Research Showcase. Instructors, more information about that's on the TEPI website. Just search for TEVRS is our acronym. We worked a long time to come up with a 5-letter acronym that didn't mean anything in any language. It was difficult, but we did it, as far as I know. And then lastly, in the pipeline coming up, the project that I'm leading is the next edition of the Tiny Earth Student Guide. So keep your eye out for that. A few of y'all are also helping with that. So thank you, Michelle, for being here and also presenting today. My goal is to have new books able to be ordered for next fall semester. We'll see how close we get to that goal. Publication timelines are always what they are. But that's what we're working on. So keep your eye out for that. So what is Fertile Ground? This started as like code name Fertile Ground, and then the name just stuck too much, so I never changed it. The pandemic happened. A lot of webinars came out of that for Tiny Earth. We did a ton of webinars in different formats, some HQ led, some TEPI led, a lot of different topics and so on. And just over the year, interest in different topics and different formats has shifted. So this is a response to kind of where we're at with where interests are, where interests have been going, what formats have worked, and so on. So this is just an evolution of what we've learned running webinars in different ways since the pandemic. Format is only once a semester is my goal. I guess that's an easy to tweak number though, if we have more or less interest. Once a semester, just so people kind of meet all at once. I'll have 2-ish presentations from Tiny Earth instructors / students about their just emerging ideas, research questions. It doesn't have to be fully formed, like polished things. It's fun to have a theme, so presentations can be loosely connected under a theme. I can come up with a theme after I have talks and go, oh, here's the connection. Today's topic is soil. But the key thing is I wanted these series of Tapas Talks to be a place where new ideas can spark and new connections can be made, thus the name Fertile Ground, right? This being the ground for a lot of fertile stuff, right? And also tying into Tiny Earth as a name. So a rebranding and a tweak to the way that we've done webinars in the past, just to make something a bit more repeatable and lightweight and easy to run. Today's presentations, we have 3 presentations from our student presentation from Isabella and company. An opportunity for Tiny Earth students to disseminate. So there is, they'll talk more about the Fine Focus, an international journal for undergraduate microbiome research. And then our TEPI presentations today, one from Josh, one from Michelle, each under the theme of soil, talking about the work that they are doing, they've been doing, their students have been doing, and so on. I won't do Q&A after each talk. I'll do Q&A all at the end in the interest of time and also in the interest of seeing connections between things, right? And instead of positioning them as all standing apart. But along the way, feel free to put questions in chat. Feel free to have questions with the speakers over chat. Feel free to answer things before or after your talk. And then at the end, we'll have a kind of open shared Q&A and open discussion. And when we get to that point, we'll decide if we want that part on the recording or not. So I'll record it either way, and then we can always kind of, you can have a conversation. I can always cut it from the recording if you just wanted to contribute but not be recorded. That's me. I wanted to keep my intro very light. Isabella's group, it's all y'all. Who's sharing the screen?
I'm sharing.
Okay, awesome. I'll turn away to you.
Okay. Hello, everyone. My name is Isabella Smith, and I am a 4th-year undergraduate majoring in biology with a concentration in microbiology and minors in health humanities and neuroscience. I'm also the president of Fine Focus, the American Society for Microbiology chapter at Ball State University in Muncie, Indiana. Fine Focus is an international microbiology research journal that provides a platform for undergraduate researchers to publish their work and gain recognition within the scientific community in all areas of microbiology and microbiology education. What makes us unique is that we are entirely student-ran from production to print, while we highlight and share the accomplishes of emerging scientists all around the world. We are only one of a couple of the chapters in the ASM of Indiana, and we are proud to be the first to publish original undergraduate research through our journal. Fine Focus is also completely free to students. There's no submission fees, page charges, or subscription costs, which makes it accessible platform to those looking to publish their work and also read current microbiology research. Our journal is published annually in both digital and physical forms, and our deadline for our upcoming issue that we're currently working on is February 1st of 2026. Once you submit your manuscript, you should expect about an 8 to 10 week review process, and then another 8 to 10 weeks for a notification regarding your publishing status. Fine Focus wouldn't be possible without the support of our external board, which is made up of educators, professors, and scientists from over 50 universities and institutions worldwide, including countries like Canada, Germany, Thailand, Iceland, and China, to name a few. They are crucial for our success within our double-blind review process, ensuring the integrity and quality of our work that we publish is all without bias. In addition to our peer review process, all articles are indexed and searchable within major databases with their own individual DOIs. Our journal is managed through the Open Journal System, the OJS platform hosted by our university libraries. We also have our own ISSN number that is listed in the Library of Congress, which adds additional credibility to the work that we publish. So to sum up Fine Focus, it is a valuable resource for undergraduate students looking for a meaningful contribution within the field of microbiology. Whether they are submitting their own research or reading our past issues, Fine Focus adds a unique space where undergraduate student voices in science can truly shine and make meaningful contributions to this ever-changing field.
Awesome. Thank you. Could you put the Fine Focus website link also in chat?
Yes,
so that it's just in chat once you've left your
Yes.
slides too. It'd be helpful for folks. And then I assume everything else you showed is also linked from there.
Yeah.
Love it. Okay. Write down your thoughts, questions, comments, etc. in chat as we go so you don't forget them. But next, I'll hand it over to Josh.
Okay. Well, thanks for the opportunity to chat about what I got to do this summer. So I'm a TEPI, a Tiny Earth Partner instructor at UW Madison, where we're headquartered, and also at Madison College. And I want to tell a bit about what I've titled New Horizons, Bridging Engineering and Microbiology in a Soil Science Summer Workshop for High School Educators. So a tale of two TEPIs. So I am one person, but I'm a TEPI at two institutions. I don't know if I'm the only one. Maybe there's other TEPIs out there that are multiple institutions, but I have the pleasure of doing it at both the university and a technical college setting, tiny earthwork. And I got the opportunity also to set up the now semesterly poster sessions, essentially the research and scholarship celebrations each semester at Madison College. That's Tiny Earth is a big part of it, but there's also other research starting to happen. Tiny Earth actually exists it's the lab component for the organismal biology class, and also there are students that I get to mentor in independent research, so one-on-one research kind of settings. So last year, the Madison College was awarded a state grant from the Wisconsin Technical College System, and it's called the Core Industry Grant, and the target or the folks who got it are part of the civil engineering technology area. So essentially what's happening is that there's a high demand in the world, both locally and regionally and nationally, for more folks to go into civil engineering technology. So these are paraprofessionals that are helping civil engineers in projects. And there's comparatively lower student enrollment for that area. So this grant is to help kind of bring awareness to the field and get more students in high schools interested in this as a career path. So the grant, the funds go toward curriculum development and professional preparation to deliver curriculum modules to high school teachers so that they could then deliver those modules to their students in their high schools. So these are local high schools, Madison area in Wisconsin. So civil engineering covers a lot of different sub areas, including geotechnical, transportation, construction, hydraulic, water resources. But the one that's particularly of relevance to us and how I started to come into this picture is in the environmental engineering domain. And specifically, within environmental engineering is the field of agricultural engineering. So this is things like farm machinery and power, or irrigation and drainage, agriculture and environment, and energy systems. So all these things are all the sort of practices that go into agriculture, that go into farming in the modern age. Okay. Now you might be asking, why would a biologist be a part of this story of this grant that was given to bring awareness to civil engineering technology? Because I am certainly not an engineer. What happened was one of the instructors at Madison College and one of the curriculum developers and grant recipients took a new position at a different institution. So I answered the call to teach one of the modules out of there are a few modules being developed, and I answered the call to teach a module on, you guessed it, soil. Which I was very excited about, and I knew going into it that I would have a little bit of a different perspective to share, given Tiny Earth's interest in, yes, animals, but also soil itself. Some of the modules, other modules included cement and hydraulics and water analysis, but the soil module was developed in part because soil itself acts as the substrate or the ground, right, that most of our construction projects, like our buildings, are literally physically on. So engineers need to know about the soil properties of the soil, such as grain size or mineral content, because it's very relevant to construction. If a building is built on the wrong type of soil, it will start to crack and erode over time. And also the the farming aspects as mentioned. For those of us other TEPIs, you might be interested, particularly interested in the sort of format of this 3-day workshop or mini course. So it was on the afternoons this August for high school teachers to come in. The soils module was on the afternoons of Monday, Tuesday, and Wednesday. There are 14 students, high school teachers in the Madison area. And the course kind of played out the combination of discussion and videos, as well as lab essays, and some data analysis. So I'll give just a little bit about what we did in the course. As far as discussions and videos, so we started with talking about what soil is and how soil scientists are in lots of different fields, including biology, yes, but in engineering, but also chemistry, hydrology, mineralogy, etc. We then developed I had the participants develop a list of reasons why soil is important to human civilization. So as you might expect, we came up with food, right, agriculture, natural resources, minerals, and of course relevant to us, some the microbes in the soil that we can use as natural products. Also carbon sequestration and nitrogen cycling, soil is useful, and water filtration, as well as sort of culture specific around the globe. Soil is very important for various cultural, historical reasons. Okay. I also had to throw in the soil versus dirt distinction that Joe Handelsman is important to point out or makes note of. Speaking of Joe Handelsman, so I used Joe's book, World Without Soil, to inform a lot of the discussion points that were included. And I actually also showed a video clip of one of Joe's talks here at the Wisconsin Institute for Discovery a few years ago. And as part of that talk, one of the content pieces was the black soils, or the Chernozem of Ukraine, and how special and nutrient and rich they are in microbial diversity, and how good they are for farming. And the participants noted, accurately so, that this talk was given about 6 months before a world event happened. Ukraine was invaded. And then it's just interesting to note that, that the nation is holding on, much like the United States is holding on to a great natural resource in their soil. The participants, a lot of them were interested in the environment and in soil. So there was a poster that one of the participants was looking at. I didn't expect it to be there, but it was. This is Soils of Wisconsin, a map of different soil types throughout the state of Wisconsin. There were some of the other instructors, the high school teachers, take students out to local community farms, including Troy Gardens here in these pictures, and have their students actually grow food, grow crops as part of their classes, which I thought was great. Another participant hipped me to a study that I was not familiar with. Maybe some of you are, but it's a 170-year, 150-year, something like that, old study in England, where they're tracking the soil health of these various plots at the Rothamsted Manor, showing how essentially farming practices like fertilization are impacting, for better and for worse, these farm plots. Someone also mentioned to me, and I saw this later on social media, that there's also, and I don't know the efficacy of these strategies, but nanoclay development, so trying to transform desert environments into actually more fertile soils. So good conversations, good discussions. Some of the things we did in the lab: we collected soil. We did soil, we looked at the attributes of soil, including the nitrogen or phosphorus or potassium contents, and the pH of soil, various soils. And of course, I had them played out and look at the microbes. So we got some counts of bacterial growth in these soils. I was excited to find out also that Madison College has a relationship with one of our regional campuses at University of Wisconsin in Platteville. They were gifted these, this was years ago actually, that they were gifted these soil samples of specifically collected to have different soil types, including sandy clay, silt loam, clay loam. And despite them being years, like almost a decade old or something, just sitting in these boxes without any water, sure enough, we plated them out, and guess what was in there? Microbes. So we got bacterial assessments from these different soil types that I've shown here in this picture. And as predicted, which was great to see, love it when sort of the data matches the prediction. In the sandy loam, we had but little growth compared to clay, and even more so the silt loam soils. Finally, we did some data analysis in the class. So we did some statistics on the data that we collected, and matched it to the samples that we collected outside, the soil type as best we could, and did some kind of conclusions about what we discovered. Some outcomes and reflections. I had them fill out a survey after we were done, and I just want to show some select responses here to get a sense of what they were feeling about the 3-day workshop. So I said one of them was, what was your favorite aspect of the course and why? One participant said, I liked learning about the various types of soil and have not thought about the soil as being problematic as it is. This is in reference to the soil crisis. I think that water and other environmental problems have been publicized more, so it was neat to learn more about soil. Now I think I can discuss soil more easily and present some of the issues about it. It was neat to see the various amount of microbes there are in each soil type. Some others here. Combination of lab and theory and lab activities was a nice balance of thinking and doing. Next was, I most enjoyed the hands-on parts of the course. It was fun to compare the data we collected as a class and analyze the differences between soil types. Lots of great conversations and time to actively work. And then learning about the soil crisis and different types of soil. And learning that we do know actually how to address the problems we have with soil. So the sort of optimistic approach that Jo presents in her book. Next question was briefly, how do you plan to implement these ideas or activities into the classroom? Participants said, I certainly could adapt some of these tests and inquiry design for a biology class. I could also envision giving some of this material to our AP environmental teacher to use. I enjoy asking my students to grow bacteria in class. I think my students would enjoy growing bacteria from soil around our school. I plan to ask the biology team at my school to consider adding a unit on soil to our bio curriculum. So that's great. And finally, I asked them specifically about a world without soil texts. So I said, we spent some time discussing the soil crisis, referencing content from Dr. Joe Handelsman's book, A World Without Soil. Please reflect on how you might incorporate this content into your classes. Someone said, I already have it ordered, and I might pull excerpts from the text for my gardening class. Another said, I intend to buy a copy of the book, and I might offer it to students who show an interest in soils, or I could find relevant passage that students could read before we perform tests on the soil. And finally, someone says, I could definitely use some of the video or content in our integrated science course when we talk about how humans are influencing climate change with our farming practices. So final thoughts. I think the important thing for me to note as a non-engineer doing, being part of modules that were created for a grant for engineering is that it goes to show how applicable the Tiny Earth curriculum and aims are, that I was able to introduce some of the lab techniques and discussion points that we do in Tiny Earth for a very new audience, or if I may open it up to a whole new horizon. So with that, I will thank Tiny Earth and Madison College, as well as the grant from Wisconsin Technical College System. Thanks for listening.
Awesome. Thank you, Josh. Okay. As always, put questions, thoughts, ideas, etc., conversations in chat. We've already had a whole conversation going about fine focus, which is good to see. I will verbalize that when we get to the Q&A part, for the sake of the video. After Josh is Michelle. Michelle, it's all yours.
Hi, I'm Michelle Korir, and I am a TEPI at Aurora University in Aurora, Illinois. And today I want to talk about my project that I titled From Soil Properties to Antibiotic Potential in Interdisciplinary Undergraduate Research Experience. And so I want to talk a little bit about how I implemented Tiny Earth into a research class and also share some of the data that we collected from what we did in this class. And so first, a little bit about the class. So this is a course called Research in Biology. It's Bio 3970. So it's taken primarily by seniors as well as a few juniors. And this is a class that is designed to be an intensive hands-on research experience. And so when we were redesigning this class, I thought Tiny Earth would fit perfectly into this to do our whole redesign of the course. And for other TEPIs, the way that this works is we have two class meetings for 2 hours each class meeting. So we have a total of 4 hours per week to do this, and it's all in the lab. And then this is also a class that we have two instructors that teach this. And so I am one of the instructors and I have that microbiology background. But then I also co-taught this with another instructor, Richard Boniak, who has expertise in soil science. He unfortunately is teaching a class right now, or else he would have wanted to also participate in a little bit of this since he was there. But instead, he just gave me some feedback on how to talk about soil. That also brings up one caveat of I am not the soil scientist, but I have learned a lot teaching a class along with him. And so for this class, a little bit of the outline of how we did this is we actually decided to have the students interact with two different soil samples throughout the semester. So the very first soil sample that we did, the students, we provided it for them so that way they could use that one to learn. So they can learn about all the tests that they did for the soil properties. And we did a big module on soil properties and then also just learning how to culture microbes from those soils. And then using those same soils, we also used a different module that's not part of Tiny Earth, but pairs really well with Tiny Earth called Pair. And I'll talk about that in a little bit, but that was where they could test antibiotic resistance levels, which adds another layer of importance for why we're looking for antibiotics in the first place. And then after they did those two modules with those provided soils, we then had them decide where they wanted to pick their own soils from. So then they can start using that. And then brought that into the lab to analyze and look for antibiotic producers. So that was just the overall outline for the entire semester. And so, first, with those soils that we provided for them, this was something that Richard Boniak had collected because he has a partnership with some of the local parks that he was able to go in and grab soil samples for that he uses in class. And so he collected from 4 different sites so that way the students could see results from a few different types of soils. And so the first one was from a local forest preserve. And so we had our forest sample. He also grabbed some soils from what was classified as a prairie, which is relatively untouched. There was some hiking trails through there. The other one was actually the university garden, so that way we could see what garden would have. And the last one we just called field generally, but it was actually a sports field. So this was where there was the grass, a lot of people running around on that essentially, but that would be our field. And then we had a total of 4 groups. So the class had 12 students in it, and so 3 students per group. And each group was given a different soil sample to start to analyze and work with. And we wanted to look at a wide range of soil properties to really get at just this initial question was how do the physical and chemical properties of the soil correlate with how much bacteria are there? More to help them decide where they would want to be able to pick their soils from later on in the semester. So that was the really big goal with this one. And so one of the things that we had them do once we gave them their soils is we gave them the lat longs for the collection site, so that way they could put that into this website called the Web Soil Survey, so they could explore a little bit of what's already known about that area. And so this is a website that you go into a map, and then this first picture over here on the left is where they basically put in their area of interest. So that was the map of that area of interest. So basically where the soil sample came from, what region. And then within that, then the web soil survey gave them a bunch of different, divisions for those different sites that had different properties within that. And so, different areas, like in terms of size, they showed the acres in there. And then in that, it told them like what type of soil was there. And so we had, silty clay here, there's some loam over there. So there's a bunch of different areas within that. And then in there was when they could see where their particular soil came from. But we introduced them to this so that way when they were trying to decide where to pick their own soils from, they could use this web soil survey to decide which region they might want to pick their soils from. So they could then, if they wanted to pick a particular soil class. And so with this, they can actually for finding their soil, the locations, they could either do like a GPS location, they could use a specific address, or just any region like some other address in the area, or just kind of pick an area on the map. So a lot of different ways to look through this. And if you are interested in the web soil survey, I have the link at the bottom of the screen, or also you can just search web soil survey for this. After they did that, we then had them look at a bunch of soil properties. And so we taught them 3 different methods of how to determine the soil textural class. We did the texture by feel, which is a really quick method that can be done in the field if need be. And it has that really quick application. We also had them do the sieve analysis so they could then separate out the pebble, sand, silt, and clay from their soils. And then we also did the hydrometer method, which is more time consuming, but it's more of, I guess, what you can consider a gold standard for determining the soil textural class, where they basically take this mixture of their soil and then use the special tool, this hydrometer, to determine what soil class it is after a bunch of math. We also had them do the organic matter, and we used the method of loss on ignition using a muffle furnace. So we basically see how much organic matter got burnt off from their soil. They also looked at their soil color using the Munsel's color charts. We had the whole book in the lab that they were able to look at. And this is the color is basically going to be influenced by organic matter or metals present. And then we also had them do pH using colorimetric soil pH test to do that. And then we also looked at conductivity to look at kind of overall health, if there was like salinity or other issues in the soil that we could then maybe potentially detect by that conductivity. And so here's just a bunch of data from that because I wanted to share a little bit of data from this. And so here were the 4 groups that we had for the soil samples. And then the main thing I wanted to really look at here first was the soil textural class section, because they did all 3 methods for this. And one thing that I found interesting is that the hydrometer and the texture by feel correlated really well. And so we could actually see some great correlation between the two, even though they are very different methods of doing basically that same thing. And so if you don't have a hydrometer or don't have availability or time to do that, then texture by feel is a great way to do this. Now, the sieve analysis did give, in some cases, slightly different results with this, and that partly is because the sieve analysis is usually better for coarser textures like the sand, but is less accurate for more of those finer, clay particles, and so that's why you can get some differences between sieve versus the other ones. For organic matter, we had a big range. So these two we're not sure if there was mistakes because that 16 to 18% organic matter is really high. That's a lot higher than what you would expect. So we're not really sure what happened here. We'd have to do some more testing on this to see if that is true. But pretty big ranges in there. Soil colors are kind of all pretty similar in terms of they had more of this yellowish brown color, so pretty consistent across that. pH, we only had the colorimetric, and so they're all the same basically, but next semester we plan on using more of a pH probe to be more accurate with that. And then conductivity values, most of them are all within expected range for most soils, so no concerns with that. And then after the soil properties, we also had them plate their samples on both 10% TSA, which I just labeled TSA here, as well as PDA, so they could see the different numbers that we get on the 2 different media types. And so overall, the pattern is that we see more colonies on the TSA, the 10% TSA versus the PDA, which isn't too surprising, just since TSA is a bit more rich. We did also notice that TSA had a lot more Bacillus mycoides. We didn't really see any of that mycoides on the PDA, so that was one of the benefits. And then we did do some correlation just to see if there was any correlation between soil properties and bacterial abundance. We don't have enough data really to do much correlation, and from what we can see, there's not a whole lot of correlation here, but mainly just want to get some more data moving forward. But we had each group present all of this data in the lab, so that way they could then come together as an entire class to decide which types of soil properties might lead to their best options for bacterial abundance, or potentially also then maybe getting some antibiotic resistance, or antibiotic producers, so that way they could then pick out their soil types. We also then looked at that pair module that I mentioned at the beginning. And so this was looking at antibiotic resistance. And so we had the antibiotics on the TSA. So this is that same data from the previous slide, the TSA versus the PDA. And then for this, they basically take their dilutions and then plate it either on TET at 3 micrograms per mil for a low level resistance, or TET with 30 micrograms per mil for a high level of resistance, and then calculate out their percent. One thing to point out is there is a wide range of percentages here across the different soil samples. Again, we only did this once, so definitely need some more repetition in there. And then one group also had an error with these plates, so we didn't get any data from that one, but very, very wide ranging, up to 70% resistance, which is a little concerning. But we definitely they were able to see that there was resistance in these soils to tetracycline. And tetracycline was chosen because that is what this pair, the prevalence of antibiotic resistance in the environment module, uses as their standard data set. So that's why we had picked tetracycline. All right, so that was what they did with their soils that we provided to them. And so from there, they could take everything that they learned to now find their own soils that they had to bring into the lab. And so we encouraged them to use the web soil survey so they could decide which region that they wanted to do. They also created their own agreement, so that way whoever was the owner of the property would sign that agreement, because they learned all about that also with that bioprospecting module. And then from there, continued on with the Tiny Earth project to look for antibiotic producers. And so with that, this is a picture here of all the different soils that they brought in. So like I said, we had 12 students, and it was kind of interesting to see just the variety that we got. So some of them have a lot more of, like, there's a lot more roots, and this one here is a lot lighter than some of these other ones. There's also some that were a lot wetter. We can see some with finer textures. And so it was really great for them to see lined up side by side all the different soil types that we had. And then we also allowed them to decide which methods they wanted to use for determining their soil properties. All of them chose texture by feel for the textural class, by the way. And then we allowed them to choose which media they wanted to use. And then when it came to looking for antibiotic production, we let them choose which safe escapes they wanted to test against to look for that antibiotic production. So they had a lot of things that they could choose on their own. And then this map here highlights all the different areas, all the sites that we collected soil from. And so this, of course, is the greater Chicago area. Aurora is down here by number 12. And I really liked to see what range of area, how much of the state basically that we were able to cover across all of our students. And then for those of you, if you ever want to make a map like this, I just used Google Maps. So you can go into my Google Maps, and I created a bunch of pins and layers to do this. So it was pretty easy to do. And then I have all the soil IDs here, just kind of more for a reference. I know it doesn't mean much to you, but on the next slide is going to have all the data for this. So big table with all 12 of our soil samples, and I have them ranked by the number of antibiotic producers we found from that sample. So all students tested 25 colonies. So they all tested the same amount. They did have a difference in our CFU per gram, but there wasn't really much of a correlation between CFU per gram and number of antibiotic producers. But the winner for the one that had the most producers was this top one at 10 within 25, so quite a pretty high level of antibiotic producers. And then pH, again, we just use the color metric, so we're going to move to something else in the future for getting more fine-tuned results. And then again, we didn't see much of a correlation between organic matter and our CFU per gram, or the organic matter versus the antibiotic producers. And then a lot of them in the area were more of the loam textural class. You know, a lot of them were silt or the silt loam mostly, but there's pretty similarity in terms of textural class. And again, all of that was by texture by feel. All right, and so just to wrap up, the main thing that I wanted to emphasize here is that the students did get this authentic research experience, and they were actually able to present at 2 local symposia in the area. And it was also really great for them to see the interdisciplinary aspect of soil science and microbiology. And in fact, we even had most of this was done in the microbiology lab where they had full PPE with gloves and lab coats and safety glasses and all of that. But then occasionally we went into the ecology lab next door where they got to touch the soil with their bare hands. And so like it was really great to see that big difference between the 2 different fields that we had. And then also after taking this class, the students were inspired to start a brand new student or club called Soil Club. And so that actually is starting this semester for the first time. So that was also pretty exciting that they got so inspired by this class. And then just a little bit of their feedback. The main thing I wanted to point out here, because I know I'm getting low on time, really is that one of them said that it was this one I really appreciated being able to doing practice with certain skills before we started working with our own samples. So they really liked having that practice with the ones that we provided them. And then they also liked how the 2 of us, me and Dr. Boniak, worked together with this project. And so thank you, and I will take any questions that you have.
Awesome. Thank you. All right. Thank you to all our speakers. Thank you all for being here. I'm going to vamp for a little bit. I would love to see in chat 2 things you would love to see in the discussion for the next 10 minutes. It doesn't have to be a fully fledged question. It can be a noun phrase. I usually tell my students, write in full sentences, not noun phrases, but you're allowed to write noun phrases now. So I'm just gonna kind of catch for the recording that doesn't see the chat what kinds of things we have in chat so far. Sarah's been lobbing up some good questions the whole time on the side to get things going, so thank you, Sarah. Asking how can people use Find Focus? John responded, you know, that Find Focus would love to be a publishing platform for undergraduate students participating in Tiny Earth. We had some follow-up questions about or comments questions about love to use Find Focus as an opportunity for students to see examples of writing. It can be daunting to have to write a thing when you haven't seen an example of what the thing is, right? And I know in like, especially writing in a science class, you as the instructor have to wear 2 hats of English teacher and science teacher. I have a master's in English, and we always have conversations about how much the English department does or doesn't support its science teachers. There's questions about how Find Focus recruits reviewers. As I'll say very briefly for Isabella, and feel free to turn your mic on and add more if you want, about they don't recruit reviewers. There's a class at BSU where students review manuscripts and help design and promote the journal and so on, but that they don't recruit outside of Ball State. Then Sarah had some questions about like the soil curriculum stuff, like bubbling up between Michelle and Josh, and like Michelle's in the committee for the next revision of the Tiny Earth, so we are working to get like the curriculum stuff into the revisions too. And just some good energy about like people would love to see some soil stuff in the book, so that's good to hear as someone leading the project to get some soil stuff into the book. Let's see, curious, not sure about how to teach soil properties accurately, curious about tests for % of organic matter, various correlations. Sarah also asked what role Tiny Earth, the database, could play in the related to soil. At the previous office hours, a note that came out that I wrote down as a to-do for me was a question of, can I see a map of all my students' work? And I went, I will write that down because it's a technical question. So maybe I'll make a button that lets you do that. We'll see. Good. Okay. More about analysis of soil. So yeah. I have questions written down, but I'll go with those that are in chat first. Let's see, Josh or Michelle, anything else you want to add about how we analyze soil?
Well, so I see there's a question here about how to do the percent organic matter. And so I can answer that question as to how we did it. And so what we basically laid out the soil on some newspaper to air dry because we had to get it down to about 2% moisture content. And then once it was completely air dried, we put it into a crucible that we weighed. So we weighed a certain amount in there. And then we put it into a muffle furnace to basically burn off all of that organic matter. And then once all that got burnt off, we waited again. And then after math, basically a series of equations, you could then determine what percent of that was organic matter based on what got burnt off. And so for the ones that were a bit high, for the ones that I had mentioned, it might have been that they weren't fully dried before we put them in there. That's one possible issue.
Josh, I saw you unmiked or unmuted.
Yeah, I just think that it, with considering the next, the update to the lab manual, I just think that adding some soil, the analysis of the soil itself, modules would blend well with the soil crisis content. I mean, we're, the importance of soil and what we know about it and why it's so important, and especially certain types or characteristics. So yeah, all good things.
Good. My kind of comment slash question was between Michelle and Josh's talks, there was a lot of collaborating with people outside of the microbiome space, right? With like engineers or like collaborating with other teachers and so on. And so I guess my broad questions is like, who else would you like to collaborate with, or do you see an opportunity for collaboration? Um, and whether it's you want to say something to them or they might benefit from you, or maybe because you just want to learn, because Michelle mentioned the learning that you get from co-teaching something. So that's my vague, like, who else do you want to connect to?
I'm actually this semester going to be collaborating with a chemistry professor at Madison College for the first time on a tiny student project, who I believe just did the training recently.
Awesome.
Mm-hmm. Kristen.
One of the things that I was thinking to kind of expand what we were doing with the project that I just presented on was also collaborating with a molecular biologist here on campus to do another version of this class where we basically take those samples and look more at the maybe antibiotic resistance genes or possible other things that we have. So more molecular testing with the antibiotic producers.
Awesome. My other comment I noted for Michelle's talk, and I'm not sure how much you did this too, Josh, of when using other like existing curricular materials, whether it's in the book or like this protocol, like just things that exist, or you weren't just using them to teach, you also kind of assess them through the process of teaching by collecting all your students' data and going, well, this one has this caveat that wasn't mentioned and so on. So I thought that was a neat thing of just the way that we, in these science classrooms, when we use things and all the students' data gets compiled, we're also assessing the thing that we're teaching from too, and go, it's all kind of a mess.
You mean there are strategies we can implement to make it even better?
Yeah, that's what I mean. Good.
Yeah, I mean, that's part of why I thought I could also help out with the revamp of the manual to add some more of those soil protocols.
Yeah. No, I'm loving the follow-up conversations here. I will definitely we have another revamp meeting coming up soon, so we'll make sure that these are part of that conversation, or on our list of things to talk about. Thank you all to our speakers. I'll start with Isabella, and then I'll just go in presentation order. Josh and Michelle, where can people learn more / connect with you, etc.? Because part of this is making connections.
Yeah, I kind of put it on the screen when I shared, but like we have a LinkedIn, we have our Instagram, and we always send out updates on our LinkedIn about deadlines, events, and how to get connected with us,
Awesome.
and
Josh?
on our website as well.
Sure. You can Google my name. It pops up first, usually under the Wisconsin Institute for Discovery. I'm under people, or just my last name at wisc.edu, wisc.edu.
Awesome. Michelle?
I just put my email into the chat, so if anybody wants that, it's there, but you can also look up my name um, with Aurora University. Biology department is pretty small, so you can just find me on the website there, and I'm also on LinkedIn.
Awesome. Thank you all. Thanks to all of our presenters, my co-host Sarah for popping in a lot of questions during presentations. Thanks to all the people who showed up. A lot of people registered this morning, and that was good to see.